Welcome. Good morning, good afternoon, good evening to all of you from wherever you are joining us today. We have had a wonderful Science Day so far, and I hope everyone is energized and excited for 3 phenomenal talks on some of our most vexing health conditions. Nearly every one of us knows somebody who has dealt with heart disease, chronic kidney disease, liver disease. All of us is a data set that is perfectly positioned to advance our understanding of these conditions and the variables that affect outcomes. And today I will be joined by 3 rising stars in the scientific world who are looking at these chronic long-term conditions using the All of Us data set. I am going to go over a few bits of housekeeping and then I will introduce our panelists. As a reminder, today is a public event, so you may always have the option to leave and change your display name. Just be mindful of what you put in the chat. There is a closed captions icon in the top right corner of the video, so please do select that if you need to have closed captioning on. And if you run into any technical issues or need an accommodation, please do email allofuscienceday@pyxispartners.co. If you want to ask a question of any of our 3 speakers, next slide, you will see a Q&A option at the top right of your screen. Please type your question there and submit it. Questions that are submitted through the general chat box will not be monitored, so please be sure that you are using the Q&A box. With that, I am delighted to introduce our 3 panelists today. We will be joined by Nicholas Panyanabong, who is a graduate student at Johns Hopkins School of Medicine, with James Hilzer, who is a postdoctoral research fellow at the University of California, Los Angeles, David Geffen School of Medicine, and with Sade Graves, is a PhD student at Meharry Medical College. So with that, please remember to put any Q&A into the Q&A box so that we can get all of those questions answered. We will do the Q&A after all 3 talks. And with that, I will now pass it over to Nick.
Thank you so much, Dr. Devaney. Hello, everyone. It's a pleasure to meet you. My name is Nick, and today I'm excited to present to you my project entitled Liver Safety, the association between the steatosis-associated fibrosis estimator, or SAFE score, and severe liver outcomes in the All of Us cohort. Next slide, please. In today's project, we took a look at the association between 3 non-invasive liver fibrosis scores, the Fibrosis-4, the NFS, and the novel SAFE score, and severe liver outcomes within the All of Us cohort. We found the SAFE score was strongly associated with severe liver events, and that it held the potential to improve the prediction of severe liver events among the population. We also observed that there was racial and ethnic heterogeneity in the strength of the relationship between the SAFE score and liver outcomes. This work is important because the SAFE score, in tandem with other non-invasive risk scores, holds the potential to offer a cheap and non-invasive alternative to liver biopsy and improve the detection and prevention of metabolic liver disease worldwide. Next slide, please. First, for some background. Next slide, please. So first, what exactly is metabolic liver disease? Metabolic dysfunction-associated steatotic liver disease, or MASLD, is a chronic liver condition that exists on a spectrum, ranging from fat buildup, or steatosis, all the way to irreversible scarring and fibrosis of the liver, known as cirrhosis. The global prevalence is around 1 in 3 adults worldwide, and the economic burden of MASLD is estimated to exceed $100 billion each year in the United States. Next slide, please. What's tricky about MASLD is that detection is very unlikely in its early stages because MASLD is largely asymptomatic. And prior studies have estimated that as many as 90% of patients who ultimately receive a diagnosis of liver fibrosis were previously undiagnosed with MASLD. One part of the problem is that MASLD diagnosis and staging has traditionally relied on liver biopsy, but liver biopsy can be expensive, it's invasive, and it requires follow-up with a liver specialist, which can all present barriers to care. Next slide. To ameliorate this problem, scientists have been working on developing non-invasive, cheaper alternatives to liver biopsy. And those non-invasive tests are designed to use routine laboratory values like liver enzyme levels or platelets or BMI, etc., to help quantify a person's risk of developing advanced fibrosis. And on this slide, I'm showing you two of the most common options. The first is the Fibrosis 4 Index, and the second is the NAFLD Fibrosis Score, or NFS. While these fibrosis scores are commonly deployed in clinical practice, their main drawback is that they're not well suited to screening in the general population. Both of these scores were developed and calibrated in high-risk, high-acuity cohorts of patients. And as a result, they suffer when it comes to low-prevalence primary care settings. In fact, one study estimated that the negative predictive values of these two scores in the primary care setting was only marginally better than flipping a coin. And thus, the SAFE score was developed to address these limitations. Next slide, please. So on the slide here, you can see the SAFE score, and I won't belabor the formula. But essentially, the SAFE score is used to bin patients into three different categories of risk: low, intermediate, and high. And what's different about the SAFE score is that it was developed specifically to optimize for negative predictive value and sensitivity, and thus to be deployed in a low prevalence setting like a primary care office. In the debut of the SAFE score, the authors found that the SAFE score had the highest area under the curve for diagnosis of MASLD, and in a follow-up survival analysis using NHANES, SAFE had the strongest association with all-cause mortality. Next slide, please. But despite these promising findings, there were still a few stones left unturned in the initial paper that inspired this project. The first aim of this study stems from the fact that the SAFE score was only validated among an elevated risk cohort, and that only associations with all-cause mortality were explored by the authors. Therefore, the first aim of this study is to quantify the association between the SAFE score risk strata and severe liver events among a population-based cohort like all of us. The second aim of the study was to determine whether there's heterogeneity in the relationship between the SAFE score and severe liver outcomes by racial and ethnic subgroup. Next slide, please. And finally, the last aim of the study was to determine whether there was concordance between the SAFE score and the existing fibrosis risk scores, and whether that concordance was informative in assessing the severity of a person's risk for liver disease. Next slide, please. All right, now on to our methods. Next slide, please. All right, to be eligible for the study, participants needed to meet a few criteria. The first, and the most self-explanatory, was that each participant needed to have sufficient laboratory data in all of us to calculate at least one instance of the SAFE score. Participants also were excluded if they had a history of significant alcohol consumption, because the study was focused on metabolic causes of liver disease as opposed to alcohol-related pathology. The third inclusion criteria was that participants needed to possess at least one feature of metabolic syndrome, so things like hyperlipidemia, hypertension, obesity, etc. And finally, participants needed to be free of any severe liver pathology at study baseline, including things like ascites, cirrhosis, liver cancer, encephalopathy, or hepatitis. Next slide, please. Now comes the part where we actually calculated our fibrosis risk scores. As you may have observed on that earlier slide, each of these fibrosis risk scores uses a wide variety of lab components. And to calculate scores for each patient, we needed to find a way of converging all of them. And so to do that, what we did was for each patient, we would go through their medical record and identify each date at which they had an ALT and an AST enzyme level measured for their liver. For each eligible date that we identified, we would then search within a 90-day window preceding that date to identify the other lab values that we needed to calculate the score, including things like globulin levels, platelets, and albumin. If we identified all of those values within that window of time, we would use them together to calculate a single instance of a fibrosis score for that patient. And we set the index date for that patient to the date of the ALT or AST measurement. And then we then stratify the patients into high, intermediate, or low risk categories for each score. Next slide, please. We used a composite outcome that was comprised of 5 different severe liver conditions that are listed here. Those were cirrhosis, ascites, hepatic encephalopathy, esophageal varices, and hepatocellular carcinoma, or liver cancer. Next slide, please. We also included a variety of other covariates in our regression analyses, including race and ethnicity, age, and sex assigned at birth. Next slide. Finally, our analysis plan had 3 key stages. The first was a descriptive analysis where we compared participant characteristics across different risk levels of the SAFE score. Then we performed a survival analysis using Kaplan-Meier methods. And finally, we did secondary analyses where we repeated our survival analysis within different racial and ethnic strata, and where we also looked at concordance between the SAFE score and other liver risk scores to see if that improved performance. Next slide, please. Now onto our results. The final analytical sample comprised a little over 100,000 total participants, the majority of whom were white and female. 4.5% of all participants in the All of Us cohort experienced at least one of those severe liver events. And the mean age at study baseline was 53.4 years old, with a median follow-up time of around 7.5 years. And on the right here, you can see sort of the breakdown by racial and ethnic group of the different risk categories. And so we can see some disparities there. For example, within our Black and African American participant cohort, we had a higher proportion of participants in the intermediate and high risk categories compared to the low risk. Next slide, please. All right, now moving on to our survival analysis. On the left here, you can see our Kaplan-Meier survival curves. You can appreciate the fact that the high risk category had an attenuated survival compared to the low and intermediate groups. And then on the right, you can see our crude incidence rates. Across all three fibrosis scores, we can see that the high risk category had an elevated incidence rate, but this is particularly true for the FIB4 group. And this makes sense when you think about it. The FIB4 was developed among a high risk cohort. It's highly specific. So participants in that high FIB4 category are going to have really bad outcomes. Next slide, please. Then in our Cox proportional hazards models, we see a similar pattern with all of the scores having significant association with severe liver events, with the fibrosis 4 in particular having the strongest association of 6.33 in the adjusted model. Next slide, please. Then when it came to our concordance analysis, we observed that the SAFE score had the higher risk assessment than the FIB4 or the NFS in the majority of discordant encounters. In other words, what that means is that in all the instances where the SAFE score disagreed with the FIB4 and the NFS, it was more often than not that the SAFE score rendered the higher risk assessment. So for example, you can see that the SAFE score classified a lot of people as high risk when the FIB4 would only classify them as intermediate and so forth. We also found that people who were high risk across all three SAFE scores had a 3.72 times higher incidence rate of severe liver disease compared to those who were only high risk under the SAFE score alone. And next slide, please. And finally, we did observe some degree of racial and ethnic heterogeneity in the association. I want to call your attention really to the high SAFE group there, where you can see that the hazard ratio for the high-risk SAFE score was considerably higher among non-Hispanic white individuals compared to non-Hispanic Black and Hispanic individuals, highlighting some degree of heterogeneity in the magnitude of the association. Next slide, please. All right, now for some discussion points. The first main takeaway is that the high and intermediate SAFE categories are associated with severe liver events in the general population, albeit a weaker association than the FIB4, since the FIB4 was calibrated for acute situations. Next slide, please. We also found that concordance between different non-invasive risk scores may improve the assessment of risk, with participants who are high risk under all scores having a considerably higher incidence of liver disease compared to those who are only high risk under the SAFE score alone. Next slide, please. And finally, we observed heterogeneity in the performance of the SAFE score across different racial and ethnic subgroups. And future studies should evaluate differential predictive accuracy of these scores and consider whether the SAFE score needs to be recalibrated. Next slide. Finally, there were some limitations of the study that warrant further exploration. The first was that All of Us was missing a clinical gold standard. So when it comes to talking about metabolic liver disease, we would ideally be able to have robust imaging and biopsy data so we can really quantify the degree of a person's fibrosis. There was considerable missingness of this data in All of Us, and so we weren't able to use that clinical gold standard in our analysis. There's also some potential selection bias. Participants needed to be alive within the past decade so that they can consent to sharing their data with All of Us. There's potential misclassification when it came to survey instruments for alcohol and smoking or lab values. And finally, we had limited power for subgroup analyses when it came to Asian American cohorts, Middle Eastern and North African cohorts, or Native Hawaiian and Pacific Islander cohorts, just due to limited sample size. Next slide. All right, these are my works cited. And the next slide, I would like to give a big thank you to the All of Us research program and all of the participants for making this research possible. Thank you.
All right, great. Well, I will start. Thank you very much for having me today. And today I'm going to discuss with you a little bit of work we have been doing in the gene environment interaction space. Next slide. First, I just want to thank everyone from all of us for allowing access to this data and for us being able to leverage this for the benefit of everyone involved. Next slide. So first, I'll just give a quick brief overview of what we did, what we found, and why it matters. What we did was we looked at the DNA of subjects in All of Us research program and compared it to satellite imagery data from NASA to see how living in areas with high air pollution could affect the risk of heart disease. What we found was that people living in areas with higher air pollution have significantly increased risk of heart disease, and identified a potential genetic switch that makes some individuals even more vulnerable to the harmful effects. And why this matters is because this research helps us understand why some people are more likely to develop heart disease than others. Next slide. So epidemiological research has identified several key risk factors associated with coronary artery disease, otherwise known as CAD. Genetic studies such as genome-wide association studies called GWASs, which use common variants, and gene burden analysis, which use rare variants, have validated many of these associations. However, despite the identification of hundreds of genetic loci associated with CAD risk, there is still missing heritability. Now, this missing heritability could be discovered through greater sequencing efforts or larger GWASs, but an equally compelling reason could be the role of environmental factors and their interactions with genetic predispositions, which are less commonly captured in traditional genetic studies. But the critical question remains, what is an environmental factor that can interact with genes to influence disease susceptibility? And this question is what led us to explore the intricate interplay between genetics and air pollution. Next slide. So previous studies have consistently demonstrated a reproducible link between exposure of air pollution less than 2.5 microns in size, called PM2.5, and an increased risk of CAD. But what is PM2.5? And to that, I'll say PM2.5 are really tiny particles that come from power plants, factories, and wildfires, amongst many other sources of air pollution. This figure on the right does a great job illustrating the complex mechanisms by which PM2.5 can influence cardiovascular health. So, upon inhalation, PM2.5 interacts with cells in the lungs, triggering inflammation, where these inflammatory mediators and secondary products spill over into systemic circulation. PM2.5 can also enter the bloodstream and directly affect cardiovascular tissues themselves. And despite this knowledge, very few gene air pollution interactions have been identified. So we address this by using large biobanks, such as, next slide, the All of Us. However, since air pollution exposure data was not readily available, we decided to use NASA satellite imaging data for PM2.5 levels. So we overlaid that data with 3-digit US zip codes made available in the All of Us for subjects. Next slide. And here you can see how PM2.5 levels were mapped. Now, to confirm we were mapping PM2.5 properly, we also used a map of domestic PM2.5 levels from CDAC to compare. Next slide. And as you can see, there is a great deal of concordance upon what we were able to map for All of Us and what was known. Next slide. Therefore, we wanted to validate previous epidemiological studies showing that PM2.5 is associated with CAD. And as you can see, in both the base and the fully adjusted model, PM2.5 was associated with risk of CAD. And in fact, these odds ratios were consistent with other large biobanks, such as the UK Biobank and GeneBank, although I don't show this data, so just take my word for it. These results established the basis for exploring PM2.5 exposure in All of Us. So, building on this foundation, next slide, we conducted a genetic analysis using a candidate gene approach. And here you can see what we call Manhattan plot, which is a visualization technique used in genomics that depicts the parts of the genome that are actually associated with a disease. And in this case, we are looking at a Manhattan plot for coronary artery disease. At each peak, there is a lead genetic variant. And these are the genetic signals that we assessed for potential interactions for PM2.5 exposure. And of these 320 lead variants tested, next slide, we identified a potential gene-environment interaction on chromosome 15 with a specific variant called RS6494488. Don't get bogged down in the numbers. This association replicated in 2 additional cohorts, and the meta-analyzed results are shown visually in this bar graph to the left, next slide, but also numerically in the table over here to the right. And I hope you can appreciate that the yellow bar graph, which represents the AA genotype, is much higher than the blue and the pink bar graphs, depicting that the meta-analyzed effect of PM2.5 exposure on risk of CAD exhibited a significantly heightened association in those with the AA genotype. This is compared to the null association observed in the GG homozygotes and the modest association observed in the GC heterozygotes. This stark contrast that you see in the odds ratios for PM2.5 exposure on CAD across these genotypes underpins the statistically significant interaction p-value shown over here on the top left of 2.2 times 10 to the negative 5. But what could be a potential mechanism for how this variant is giving a statistical signal? Next slide. To unravel a potential mechanism, we used a StarNet cohort to conduct an expression quantitative trait locus analysis, which is one way to identify which gene might be implicated at a locus. Now, interestingly, TRP4 was the only gene whose expression was consistently seen to be decreased in cardiometabolic tissues as a function of carrying the A allele. Since the A allele is the risk allele, it suggests that TRP4 expression is protective against CAD. This directionality is reinforced from looking at additional data from StarNet, which is presented in the green box plots in the center. And you can see that TRP4 expression was significantly reduced in atherosclerotic aortas of CAD patients compared to their controls. Now, given the pathophysiology of CAD and the fact that endothelial cells are in direct contact with the arteries, we hypothesized that this expression of TRP4 could be modulated in this cell type. Next slide. Which we confirmed through looking at single-cell RNA sequencing data, which is a way of determining what cells in a tissue express a certain gene. And you can see that TRP4 is robustly expressed, shown in the purple, in a majority of the cell types within atherosclerotic plaques. So therefore, we interrogated a population of endothelial cell lines sourced from about 150 multi-ancestry donors, which also showed that the A allele, or individuals or subjects that had the A allele, had decreased levels of TRP4 expression. Next slide. So to investigate whether TRP4 expression was responsive to air pollution as well, human microvascular endothelial cells were also exposed to diesel exhaust particles, also called DEP. And we show that TRP4 expression was significantly reduced after DEP extract exposure. Next slide. And to further validate these findings, we conducted an in vivo murine experiment where mice were chronically exposed to standardized form of DEP for 8 weeks. And consistent with our human data, TRP4 expression has significantly reduced expression in the aorta of exposed mice. So these findings strongly reinforce the role of TRP4 as the effector gene in this pathway. Next slide. So taken together, this data suggests a biological model wherein genetic susceptibility due to the A risk allele at RS64944488 leads to decreased TRP4 expression in vascular tissues, which increases the risk of CAD. And in parallel, PM2.5 exposure further suppresses TRP4 expression, which synergizes with this genetic repression to amplify the overall risk of CAD. And next slide. With that, I'd like to thank our collaborators spread out over the US. I'd like to thank the PIs, Human Alai and Yana Hartiala. And that is the end of this presentation. So thank you very much.
And please keep adding questions so we can have a lively Q&A session right after Sade finishes.
Hello, everyone. My name is Sade Graves, and I'm a student at McHarry Medical College in the Biomedical Data Science Department. And today, I would like to discuss exploring heart failure risk prediction in dialysis patients, combining clinical data and social factors. Next slide, please. Why does this matter? Heart failure is a major complication for patients with chronic kidney disease who depends on dialysis. And we know that 37 million US adults have chronic kidney disease, and 9 out of 10 actually have early stages of chronic kidney disease and don't know it. These patients who progress in dialysis, their risk of heart failure actually rises very sharply. Next slide, please. For our study overview, our projection was to build a predictive model for heart failure using electronic health records, plus using social factors of health. We asked ourselves 3 main questions for this study. The first was, do social factors like income, marital status, employment, and education actually affect heart failure patients who does dialysis? And the second is clinical factors like diabetes and hypertension, are there the main drivers? And the last question was, the social and clinical factors actually interact in predicting heart failure? Next slide, please. So in order to build this cohort, we actually use our data from all of us using a retrospective data set by combining clinical data as well as social survey data. And the clinical data that we combined was end-stage renal disease diagnosis, as well as chronic kidney disease diagnosis data that those who were dependent on dialysis. Using that, we also combined their core morbidities such as hypertension and diabetes, and we looked at demographics such as their sex, their age, and their race, using electronic health records. And we obtained the social factors of health data using the basic survey that's in all of us, looking at education, marital status, and using the start date time of when they first diagnosed with chronic kidney disease, as well as the start date time when they actually did dialysis treatment, as well as when they were diagnosed with heart failure. Next slide, please. In our methods, our outcome that we wanted to look at was heart failure diagnosis after dialysis, and we used 2 models. We applied 2 models for this: logistic regression for association and odds ratio, and gradient boost for prediction and feature importance. We handle missing data with multivariate imputation and check multicollinearity with VIF, and addressed it imbalance with smoke, and we validated with the stratified split. Also, just to correct an error, that we actually looked at the AUC, the area under the ROC curve, not accuracy. Next slide, please. As we built the final chronic kidney disease dialysis cohort, our main number was 23,429 patients. And out of those patients, 12.4% were diagnosed with heart failure after dialysis treatment. And out of that same cohort, 39%, over 39% had diabetes. And out of those who had diabetes, 17% had heart failure. And also those who in the same cohort that actually was diagnosed with hypertension, 63%. And out of those who had hypertension was also 16% were diagnosed with heart failure. I want to please note that there were some patients that actually had hypertension as well as diabetes diagnosed at the same time. We did exclude for our model 11% of those who had heart failure before they did chronic kidney disease dialysis treatment. Next slide, please. This slide displayed the breakdown of our demographics. And one thing I want to highlight that majority of our patients were white, non-Hispanic. However, what's significant was the Black or African-American descent had 17% had heart failure. That was very significant for that population. And the lowest we had were Asian descent, had over 8.5% had heart failure. And males had more, had over 13% heart failure compared to females. Next slide, please. This slide actually displays our cohort's social factors of health, and we actually looked at the four main factors that we want to look at. For example, with income, most of our income groups were grouped between 10,000 to 35,000. However, in that same category, the highest heart failure rate actually came from the lowest income group under 10,000. And for education, majority of the patients had either 34% had over college or beyond, and 11% had less than a high school. However, the most highest heart failure rate came from those who had less than high school education. And for employment, 75% were unemployed. However, those who were employed significantly had the lowest heart failure rate of 7.7%. 7.7. And with the marital status, majority of the patients were married of 41%, and some were also, and those who were married or never married or widowed, the highest heart failure actually came from those who were divorced patients, over 13.5%. Next slide, please. The model that we used, the first model that we did was the grading boost, and just to look at the top features and the AUC score of that, now accuracy was, it predicted more high risk from the low risk patients when it comes to heart failure, and the most important features were the hypertension, age, diabetes, race, unemployment, and income. Next slide, please. And well, logistic regression, our AUC was 0.88%, and this showed the association between the characteristics of the variables of the social factors of health as well as the demographic and the comorbidities. For example, hypertension, those patients who had hypertension were twice as most likely to have heart failure compared to those who had higher income, had lower heart failure risk, and those who were unemployed actually raised their heart failure risk odds by 42%, and those who are diabetic also had an increased heart failure about 44%. And it's just our previous slide for the demographic of those when it comes to race. Even with this association, it shows that 75% of those who are Black race compared to non-Black had 75% more of a higher risk of heart failure. Next slide, please. In conclusion, these findings kind of highlight the importance of combining, back to our research question of, is it important to combine clinical and social factors just to understand risk prediction or intervention, and understanding also the early heart failure diagnosis occurs also before dialysis. However, it will also be able to identify certain other factors that affect patients when it comes to that. Future work, we would like to incorporate more medication diagnosis as well as lab work. Next slide, please. I would like to sincerely I want to thank all of us research program and all the participants who made this possible, those who volunteered to give their information to all of us and allow certain researchers and students like me to be able to explore this rich data set. Next slide, please. And I lastly like to acknowledge my mentor at my school and my mentor, my school, all of us, Science Day, as well as my institution of McCary Medical College. Thank you.
And thank you to Nick and James. I'll ask everyone to join me back here for some Q&A. We got a lot of really great questions. Sade, I want to start with you first. You said at the end that one of the things you'd consider for future work might be to incorporate vital signs and medication data to improve model performance. Can you say a little bit more about what you're thinking there with respect to, in particular, medication data?
Especially like common blood pressure medication, whether from personal experience, like certain medications that dialysis patients do take, to see some medication they give you for your heart failure, and just look at that, or well as certain medication of the insulin they may take. I would like to look at that as well.
Wonderful. Thank you so much for highlighting the social contributors to health outcomes. It's exactly what we are hoping this data set is going to be useful for. James, speaking of contributors, the environmental impact, genes by environment research has been of wide interest for a long time now, because we all know we have our own bodies, but it's also the context within which we live, right? Both the social factors that Sade highlighted, but also the environment. So a question for you in particular with the PM2.5, is there something unique about coronary arteries and PM2.5 exposure that causes disease there, or do you expect all vessels, including cerebral or peripheral and vascular, would have increased disease as well?
Yeah, that's a great question. So we're actually working with a few endothelial cell types to determine actually their response to air pollution and see in the context of what type of air pollution, like PM2.5, there's even smaller air pollutants that could really permeate and maybe cross the blood-brain barrier to get into that sort of system over there. So there's a lot of research that can go on to this. As speculation goes, I might say that we're trying to figure out if that's the case. So that area of research is currently ongoing, and I think that it's reasonable to think that it's possible.
Excellent. And while you have the mic, do you think that your findings would be different if you were to have access to more granular location data from all of us? Which sort of gets to a question I was going to ask. That came from an attendee, but you mentioned using the NASA data set, and I was wondering if there are other data sets that would also help expand the work. So I think those two questions kind of go together.
Yeah, definitely. Okay, so I'll go ahead and start with the latter and then work my way to the former and say there are a lot of data sets out there that are not just NASA with PM2.5, but NASA has a lot of other air pollutants that you're able to essentially leverage. Now, to the former question, which is, the granularity really depends on where the data comes from and what you get the data for. So NASA is really able to get highly granular data. So you would assume that if you were able to get finer resolution on zip codes, you can integrate that with the very granular data of the NASA, basically the data files that they provide you, to give you a little bit more nuanced understanding. So like if you're looking at it in the context of 3 area zip codes, I mean, you could see from the map that I showed before, but those are pretty substantial regions. So it would be really nice to be able to get specific sorts of areas. And then that would also help when you're talking about like regional biobanks as well. At UCLA, they have one called Atlas, where all these individuals live in like that 3-digit zip code. So you're not going to really get granularity at that point when you're looking at smaller ones. So it would be really nice to be able to leverage that sort of 5-digit zip code, and as well as other air pollutants, as you bring up, Stephanie.
Yeah, thank you. I don't know if other attendees are feeling this way, but I'm now very curious about the PM 2.5 levels in my area.
Yeah. Well, shoot me an email, send me your latitude and longitude, and I'll get you that info.
Excellent. I can send that from my work and my home, and we can see which one is more safe.
Yeah, exactly.
Nicholas, a question for you. Are there other exposures outside of alcohol that contribute to the risk of fibrosis? As all of us, in particular, adds more environmental data, can you envision adding exposures to improve the risk model that aren't currently available?
Oh, totally. And actually, I've been feeling very inspired by Dr. Hilser's wonderful presentation about all the environmental exposures. I guess on the topic of environment, there are, I think, quite a few associations with liver fibrosis. So sort of the usual suspects, right? Like heavy metals, PFAS, things like that. I don't know about PM 2.5. Maybe we'll have to see and explore that one. And I think even beyond the environmental domain, I mean, things like, genetic ancestry. There's a few like high risk alleles that have been identified too. And so I could totally envision, as sort of all of us continues to flesh out those particular domains, I could totally envision factoring those into the risk equation as well.
One of the best things for us on the All of Us side about Science Day is figuring out what researchers need to help complete the studies, right? We all get a glimpse of your research midstream. You've got conclusions, but we all know there's next steps beyond. So that's very helpful. And Nick, I'll ask you one more while you have the mic. For the liver study, could there be selection bias as only people who had blood tests for higher liver risk, albumin, et cetera, were included? And therefore, there could be selection bias for patients at higher risk, even within this population data set.
I think that's totally true. I mean, and I think that is honestly just an unfortunate byproduct of like any longitudinal study that is really using like the EHR as the basis for like indexing folks and following folks up, is that you're going to be getting higher acuity patients. I guess what I would say though is that I feel like since our study is sort of comparing like patients who are like had a low risk score to patients who are at a high risk score, I mean, these were all people who were still captured in the data set. So I wouldn't anticipate, even though the selection bias exists, I don't anticipate that it would significantly skew the results in one direction or another, since sort of the comparison that's going on is sort of within the cohort, within everyone in this EHR system. We're comparing people who fell into one risk category versus another. So if anything, I think actually the fact that we have folks who are maybe higher acuity could actually kind of blunt the association, right? Because then that would mean that people in the low risk group were more severe compared to the other groups as well. So yeah, I guess in short, yes, I think that selection bias is definitely a consideration, but I don't know how much it actually skews the results away from the null hypothesis.
Excellent. Lots more for you to study. I'll ask the last question to Sade. Sade, what was the most surprising thing that you learned as you were doing this research?
How significantly social factors actually impact the well-being of those who have chronic kidney disease and dependent on dialysis.
Well, thank you all very much for taking the time to do this research, for helping us make this data set better and better, and for applying real health outcomes to people's lives. I can speak on behalf of everybody watching here. It was tremendously insightful to hear from you all. And it already, like, just from my perspective, I was able to draw these findings into what it might mean for myself and my family and my friends and loved ones. So thank you for helping draw that connection between the participants and what they donate and researchers. I am going to wrap us up and just let you all know that there will be a short survey following this session. So please complete that survey. It helps us know what works and what doesn't work about Science Day, so that every year for the many decades going forward, it gets better and better. And then after you do that, please join us for the participant forum and closing session. Do not miss it, because there will be an award ceremony. So thank you all. Thank you, Sade, James, and Nick. It was wonderful to spend time with you. Bye, everyone.